Good morning, everyone. It's a pleasure to be hosting this last talk before the summer break in this incredible IDMP seminar series on COVID-19. And I'm extra proud today to be featuring work that's been going on in the Sabeti lab and with their extensive network of partners during this time. In particular, we're going to hear from Jacob Lemieux, an MD and a PhD who's been doing a postdoc in the Sabeti lab for a while now, but is actually based as an ID clinician at MGH. And Cameron Myhrvold, another wonderful postdoc in the lab who's been driving a lot of the diagnostic and detection work that we'll hear from following Jacob. And I'm just going to give a little bit of background to set the stage for this and then hand it over to Jacob and Cameron to tell you about the work. So I think it goes without saying that genomics have become really a fundamental part of how the world responds to outbreaks and obviously the COVID-19 pandemic. And in particular, genomic data from viruses collected from patients who are infected can help us detect and identify what the causative agent of the disease is, can help us track the spread of the virus through populations, through communities, through countries and around the world, and then can serve as the fundamental information that allows us to develop diagnostics and treatments and vaccines to control and hopefully eventually end the spread of the virus. So a lot of the work that we do is actually based on phylogenetics, understanding the evolutionary relationship between the viruses that cause disease, and in this case, SARS-CoV-2. And just to orient you, I think you're going to see a lot of slides showing phylogenetic trees, which is the kind of the data type that we ultimately build our inferences on for understanding the epidemiology of SARS-CoV-2 and of COVID-19. And this is just a little toy example to show you how this works on the left, and that we can take the virus from cases, from infected individuals, sequence the genomes of those viruses, and then use that information to understand the evolutionary relationship between the viruses. And I want to clarify that that doesn't actually mean that these viruses and these patients or these cases are connected in a kind of direct contact tracing kind of one-to-one connection, but it gives us an understanding of how the viruses relate in their kind of evolutionary history. And because we can collect the samples and the data in near real time, we can infer something about how those cases are actually connected in the real world. And in fact, it's information like this that helped us identify, or helped our colleagues identify that SARS-CoV-2 was the virus causing a disease identified as a pneumonia-like illness originally in China in late 2019. And on the right, you see that phylogenetic tree showing the SARS-CoV-2 in red and how it was, in fact, a novel beta coronavirus with similarity to other beta coronaviruses that have been known to cause similar symptoms and epidemics previously, including SARS-CoV and MERS-CoV, but that this was a new beta coronavirus that had emerged originally in China. Many of you probably know about the Sabeti lab and our work that has largely focused it in West Africa, and the idea of bringing the power of genomic data and these types of tools that can emerge from it to viruses of epidemic potential in that region. And I just want to share this new kind of project that we're embarking on with our colleagues in West Africa called Sentinel, that's been funded through the Audacious Prize. And just the irony that this project was launched, the prize was announced just in March, where our proposal to the Audacious program was to build a global surveillance network to stop the next pandemic. But of course, as it turned out, the next pandemic at the time was already upon us with COVID-19. And early on, I think a lot of our thinking was that our work would focus on making sure that our partners in West Africa were protected from the threat of COVID-19, following the toll that the Ebola outbreak had taken on these communities and the ongoing threat of other epidemic viruses in the region. But our work has not only focused in West Africa. In fact, we have long worked to build the same type of genomics capacity at home in Boston, in Massachusetts, in New England, and increasingly across the US. And we're really proud to partner with the Massachusetts Department of Health and the CDC to bring training and capacity building to our own colleagues in the region, to build their capacity to do genomic sequencing and bioinformatic analysis, phylogenetic analysis, to support their public health strategies and interventions to control COVID and other infectious diseases previously. So over the past few years, we've trained folks from 7 state departments of health and planned to add 5 more this summer, with a focus on West Nile, as we thought it would be in probably around January of this year. But of course, all of that work has pivoted to COVID. And really trying to build capacity to do this locally within the states, within the state public health labs, but also to kind of backstop that with the support of the capacities that we have at the Broad to provide sequencing and analysis to support the public health efforts. So just backing up a little bit, it's just amazing to think that in February, we thought it seemed probably appropriate to apply for supplemental funds to the NIH to do some work on SARS-CoV-2 genomics, and solicited a letter of support from the head of the state lab at the Massachusetts Department of Health, Sandy Smalley, where she shared in her letter that they had 1 confirmed case of COVID-19 in Massachusetts at the time. And we were really trying to guess whether the work we would do would focus domestically or would really be trying to support the global effort. But of course, as it turned out, COVID hit Massachusetts hard. As of just yesterday, there were over 107,000 cases and over 8,000 deaths in the state alone, and we all know that the impact that that's had on our own lives and on our communities and on the country. And so we're just really proud to have been able to partner with our colleagues at the Department of Health and at Mass General Hospital, where we also have a longstanding relationship, bringing genomic tools and data to understand clinical impacts of viral infections. And over the course of COVID-19, this has really extended to include a strong partnership with the CDC Spheres network that's trying to expand genomic approaches to COVID-19 nationally, and also working closely with our colleagues in experts in epidemiology at the Center for Communicable Disease Dynamics at HSPH, and at UMass Medical School in Worcester, and extending to include other state departments of health, including Rhode Island currently, and others coming online. And before I hand over to Jacob, just to tell you a bit more about what we've actually done with these partnerships, the data that we've generated, and the inferences and impact that it's had on our understanding of the spread of SARS-CoV-2, I want to just give a big shout to our partners at the Broad in DSP, the Stanley project managers, and OSRP, who have helped us with navigating the IRB processes in real time. We couldn't have done any of this work without them. And so hand over to Jacob, and then on to Cameron to tell you how we've used the genomic data then to inform diagnostic strategies that are still desperately needed going forward. So with that, I'll hand it to you, Jacob.
Okay. Great, thank you for that introduction. As Bronwyn mentioned, my name is Jacob. I'm in the Division of Infectious Diseases at Mass General Hospital and a postdoc in the Sabeti lab. And I'm honored to be here representing our whole group. It's been an extraordinary team effort to talk to you about SARS-CoV-2 genomic epidemiology in Massachusetts. I just want to take a step back and review what seems like eons ago in pandemic time. The first case came to Boston in the end of January. It was a returning traveler. The individual presented with typical symptoms of COVID-19, was isolated, contacts were monitored, and things were relatively quiet. There were no secondary transmission episodes identified. Things were quiet through the month of February, as far as we thought. There were some, in the beginning of March, several travel-related cases related to travel in Europe, and then things really exploded in the first week and second week in March. One widely publicized event was a conference hosting international attendees, from which several individuals developed symptoms and were subsequently diagnosed with COVID-19. You can see the epidemic curve here for the second half of March and April. The epidemic peaked in the state in the third week in April, and it really just took off exponentially. We converted the Sabeti lab really into a production facility with a lot of help and incredible support from the Broad and an extraordinary team effort. Into a production sequencing facility, and developed and validated a pipeline for generating high quality SARS-CoV-2 sequence in a matter of weeks. It's a fairly standard Illumina-based pipeline, but it works at scale, and we're able to do the sequencing on a high capacity instrument, do it all in 96-well plates, and then do a lot of the analysis and assembly in an open fashion. Allows us to share the data collaboratively, openly, and all the analysis pipelines in the cloud. So here are just some pictures of extraordinary efforts in the group, including technicians, staff, graduate students, postdoc, all kind of an all hands on deck effort to generate these genomes. We're very happy with the way this works. We generate high quality sequence from approximately 2/3 or a little over 70% actually of sequences that we study. And the probability of getting a complete genome depends strongly on the amount of input starting material, but for the lowest input ones, we've also developed some other methods that allow us to generate complete genomes from those as well. And we share the results as quickly as we can. We've set up a website and a portal and linked to a working manuscript. Here are the links. And we share the data as soon as we can, which means not quite just as it comes off the sequencer, but as soon as we're able to certify that it's accurate and have some sense of the inferences that are available from the data. So I'm going to tell you today about what's data release 2. Data release 1 is up there and went out at the beginning of June. Data release 2 is about 1,000 genomes. Of those, 770 are greater than 98% complete. Those are the highest quality genomes. Approximately 3/4 of those are from Mass General Hospital, and we've had an extraordinary collaboration with the MGH Microbiology Lab and other colleagues in the Division of Infectious Diseases during the pandemic. As well as with the Department of Public Health and the Boston Healthcare for the Homeless program. Genomes from the Boston Healthcare for the Homeless program comprise about 1/4 of the cohort. We've made an effort to exhaustively sequence all available genomes from the first week to 10 days of the pandemic, because those are the most informative about the introductions into the state. As well as outbreaks at a nursing facility and sampling from one of the outpatient infection clinics in Chelsea, which has been the hardest hit area of the Boston area. So here is how the cumulative number of cases that we sequence relative to the reported cases statewide in blue. What you can see here is that we're really tracking with the reported cases for the first week to 10 days. And then over time, the fraction of cases that we sequence from the total reported of the epidemic declines as we can't keep up with the epidemic in terms of the number of genomes that we've been able to sequence. But we have almost complete coverage of the early events related to the pandemic. And the middle, we're sequencing about 10% of the total reported cases, and it drops to about 1% in the end. Because many of the genomes come from Mass General Hospital, where they're linked to medical records, we can pull from the EMR, the medical record, both clinical information and demographic information. And so we can see geographically where these cases come from. What you can see here is that we're fairly well represented in the eastern part of Massachusetts and the greater Boston area, which is accounted for approximately 80% of the reported cases. Our sample is most concentrated in Chelsea, Revere, and Everett, and East Boston. These have been the hardest hit areas of the state. So while not a random sample from the epidemic, the sample is fairly representative of what's gone on in eastern Massachusetts and in the hardest hit areas of the state. We can compare the Massachusetts data to global genomes. There are several thousand, actually almost 60,000 of these available right now, and construct a large phylogenetic tree showing the ancestral relationships among viruses. What you can see is that our genomes from Massachusetts are distributed throughout the tree, but the upper part of the tree, particularly the branches from Europe, are overrepresented in our data. And there are some places, particularly this clade here, which seem to be highly concentrated and almost private to Massachusetts. I want to emphasize that our data is publicly available and do a quick demo of this website, Auspice.Broad Institute, that we've set up that runs this incredible software called NextStrain from our colleagues at the Fred Hutch. And you can actually interactively explore these trees. You have the phylogenetic tree at the left, you have a map at the right, you see a genome diversity data where the SNPs fall underneath. And you can click on the branches and it will tell you the inferred ancestral relationship of these inferred nodes. You can zoom in, and the other thing that you can do is color by covariates and other things down here below. So if you're interested, for example, in cases in the nursing facility, you can click by that covariate and then you can zoom in on the clades that correspond to these clusters. So it really is very exciting to explore the data at that level, and I would encourage you all to do that if you have a chance. So as mentioned, the Massachusetts genomes are concentrated in the upper part of the tree. Most of the viruses that are circulating in our cohort are descended from those with European ancestry. We can use this to infer the location and in the ancestral location of viruses circulating. And we find that in the early part of the pandemic, a larger fraction were imported from outside of Massachusetts. And then that drops off pretty dramatically as the pandemic the epidemic in Massachusetts takes hold. The black line here is a logistic regression fit showing a decreasing probability of importation through the study period, and the dashed line is a smoother. So what you can see is that things drop off quite dramatically during the month of March. And by the time April hits, there's very little importation. It is concerning, and I'm not sure if this is a real trend in our data, but I think it's a real trend in general, that the probability of importation does seem to be picking up. I think as our local epidemic comes under control, we're probably going to see a greater fraction of imported cases, and we hope that those do not launch large outbreaks going forward. The association of viruses related to spread at this academic conference in late February was a key feature of the reported early events. And we do see this in the phylogenetic data as well. So the light blue dots here are cases with a known association to the conference, and they all fall in a single clade. This clade is defined by a single variant, C2416T, and it is the most recent common ancestor of all of the conference-associated viruses. C2416T appears to be a single introduction from Europe in sometime mid-February, and quickly skyrockets to over 80% allele frequency in our data. This was, and then ultimately levels off to about 40% of reported sequences in our sample set. This was important feature of the ultimate pandemic, the epidemic in Boston, in part because it took place so early. You can see the statewide reported cases all of this spread, massive amplification of C2416 took place in the early part of March, when there was not widespread appreciation that SARS-CoV-2 was circulating in Boston. There was not widespread appreciation of asymptomatic transmission, and there was not widespread use or availability of masks for the general public. And this is consistent with reported data from the state documenting that approximately 80% of known cases at the time were linked to the conference in the early part of March. I want to really be clear about what we can and can't say here related to this variant. So it is the case that all of the conference-associated cases in our data set harbor C2416T, but I want to emphasize it is not necessarily the case that all C2416T viruses are associated with the conference. We can get a little more granular by looking at or we can get more confident in the inference by looking at a variant that occurs during or after the conference only among conference attendees. And we see this variant circulating at intermediate frequency among a subset of conference attendees. So this can serve as a sort of a lower bound for the effect. And it's clear when you look at C26233, which comprises approximately 20% of sequences in Massachusetts through the sequences in our sample through the epidemic period, that this event had a major impact on the ultimate course of the epidemic. C26233 doesn't solve all the issues because it's not a particularly helpful lower bound in the sense that we know there are many individuals who are associated to the conference that do not have this. But I just want to emphasize that we have, that there's uncertainty and it is not necessarily the case that all C2416T harboring viruses are linked to the conference. And I further want to emphasize that because an event amplified transmission does not mean that it is responsible. And I want to draw everyone's attention to the early part of the outbreak. And there was a great misunderstanding that this was a single episode of emergence, crossover from animal to human reservoirs in the Huanan seafood market in China, in Wuhan. When the cases were reported from the China CDC, it became clear, and I think was shocking to many people, that the initial cases had nothing to do with the seafood market. And clearly SARS-CoV-2 was circulating well before the recognized outbreak at the market. So we really need to be very cautious about the inference that we draw. New data can really change the way that we think about the way things emerge in a particular area. But regardless, C2416T has been an important part of the global epidemic. We do see the allele exported to other countries. At the end of the conference, which is denoted by the black line, we see that there's an exponential rise in allele frequency, and it accounts for approximately 1.6% of sequences in global databases. We study other clusters. We studied a nursing facility in detail. Over 90% of the individuals in this facility, which had a major outbreak, were infected by a single clone. There were 3 introductions into the facility, but over 90% of the infections were from a single clone that predominated. This was also the case among the homeless individuals studied, multiple introductions into that population, but repeated clonal amplification within subpopulations in that group. We are just scratching the surface. There's a lot of diversity by geography. You can see this is the conference-associated allele at the top. This seems to be sort of circumferentially distributed around Boston. And then another allele, C1059T, is more in the north of Boston, and this represents a separate introduction from European sources. We investigated 2 potential outbreaks with the infection control unit at Mass General Hospital, and it is definitely not the case that every outbreak has massive clonal amplification. We actually refuted nosocomial transmission because it was clear in both of these cases, which were suspected outbreaks in nosocomial in single hospital wards, that these represented multiple independent introductions and that they or phylogenetically inconsistent with a single amplification event, a single cluster. We can use the genetic data to infer the timing of these events. When we look at the estimated time of most recent common ancestry, we see in particular that this major 2416T clade evolved, the mutation evolved around mid-February. So the data do not strongly suggest cryptic transmission prior to February, although there's one clade here where we have substantial uncertainty. So I would say that our data do not suggest cryptic transmission prior to February, but neither do they exclude it. We can also look at co-infections, and we found approximately 1% of individuals have co-infections with rhino and enterovirus. We confirmed these with the BioFire, and of the 12 that we tested for confirmation, the identifications were accurate in all of them. I want to close by emphasizing a couple of things. For those of you who have interacted with this disease clinically, you will know that this is an extraordinarily complicated disease involving an initial period of viral replication and then a subacute, prolonged, in some cases, inflammatory, exuberant, inappropriate inflammatory response. And this may involve multiple organ systems. So this is an extraordinarily complex disease. It's not just a bad flu. And we're beginning to merge the clinical data linked to these isolates with the genetic data from the isolates to do viral variant association studies. Here is one, it's an exploratory analysis linking virus load to the genome sequence. And the top hit is a variant that's attracted considerable interest from others. It's a substitution in the spike protein, and there's emerging evidence that this has led to increased transmissibility. In particular, our close collaborator, Jeremy Luban, has been leading the way and has recently shared on bioRxiv a preprint showing the cryo-EM structure and the impact that this substitution has on the structure of the spike protein. And I finally want to just emphasize the degree of extraordinary support that we've received from the whole Broad Institute and the data sciences platform in particular. And this has catalyzed a transition to us to the Terra platform, which has just been an incredible resource for launching reproducible workflows and sharing them with the public so that other people can see exactly how we reproduce and do the analysis that we do. And I think this is just critical where there's rapid open sharing of results, but people need to be able to double check those and look at the data directly themselves. Otherwise, there's no way to validate things as the data are emerging. So we're really proud that our analysis is open and it's a featured Terra workspace, and anybody can look at how we've done it and take a look at that. So with that, I'm just going to close. I just want to again emphasize that I'm honored to be presenting on behalf of a large group of people. It's really been an extraordinary effort involving many individuals in the Broad Institute as well as outside the Broad Institute, and of course recognize Pardis, who's been the driving force for all of this for a decade now. Thank you.
Thanks, Jacob. I think we'll hold questions till the end. I'm going to pass it over to CARMEN now to tell us about his work on him and his team's work on developing new approaches for SARS-CoV-2 diagnostics. And please do submit your questions through the Q&A button, and we'll get to those at the end of CARMEN's talk.
All right, can everyone see my slides okay?
Looks good.
Great, perfect. So, hi everyone, I'm a postdoc in the Sabeti lab. I'm going to tell you today about the work of quite a large team of people who work with me in Pardis's lab, and also briefly mention that in January, I'll be starting a new position at Princeton as an assistant professor in the Department of Molecular Biology. So the problem I think here is pretty obvious. We need a lot more COVID testing, especially if we really want to reopen the world, whether that's in a school, at the workplace, at a stadium. I think our institute has really led the way here and shown how important testing is. And you know, whenever I come to work at Broad, I'm really grateful for our ability to test people regularly and have the confidence that everyone I'm interacting with on a day-to-day basis has those negative test results that show that they are not, you know, potentially gonna infect someone else with the virus. And what does that really mean? It means we need tests with a fast turnaround time and tests with a large batch size, so we can test lots of samples at once. And I think, you know, depending on the scale of the problem, we really want both of those things at once, and it's really hard to do both. So in order to try and achieve this, we took a CRISPR-based diagnostic called SHERLOCK that many of you might be familiar with, developed right here at Broad by our colleagues in Feng Zhang's lab, and really tried to simplify the process into a single reaction that combines the amplification step and the Cas13-based detection step. And so that was quite a process that we've described our work, I should say, in a bioRxiv preprint. So those who are interested in sort of checking it out in more detail, I would encourage you to go there. This process took a lot of optimization. So before we had optimized the process, moving from a two-step reaction to a single-step reaction resulted in a loss of sensitivity of about 4 orders of magnitude or so, as you can see on the top part of this slide. But fortunately, we were able to optimize the reaction substantially, changing around a lot of the components in order to get the sensitivity to be similar, maybe not quite as good, and that's still something we are actively working on so that the method can be as sensitive and as fast as possible. And in doing so, we really thought about a lot of the other parts of the process too, because that sort of part of the reaction is really important, but it's really only the middle part of a workflow that goes ultimately from a patient sample to a readout. And here, what I'm describing to you is our integrated workflow that we call SHINE, which has a couple of different acronyms for it. One of them that I like quite a bit is SHERLOCK and HUDSON integration to navigate epidemics. And in particular, the way that works is we start with either a swab or a saliva sample, and we perform a method called HUDSON that we developed and published 2 years ago that allows us to use a combination of heat and chemical treatment to rapidly inactivate nucleases that are present in the sample, as well as viruses themselves. And then the output of that reaction can be put into this single-step SHERLOCK reaction that I just described to you. And within 30 minutes to an hour, that reaction will proceed, and then you can do a readout, which is either a fluorescent readout inside of the tube, and that's something that we've really focused on quite a bit recently with a companion smartphone app to take a picture of that tube to report the result, or you can do a colorimetric readout on a paper strip. And depending on the circumstances, you might prefer one or the other of those readout modalities. In particular, one of the things we were able to show in this work is that this method is compatible both with nasal swabs and saliva samples. So I'm showing you here what are essentially limit of detection experiments performed in either the swab media or in saliva, where we are diluting an RNA target to different concentrations and testing that out. And what you can see here using our in-tube fluorescent readout is that the limit of detection is somewhere between 5 and 10 copies per microliter. And here the total time from sample to answer was about 1 hour, 70 minutes. So this was 1 hour of detection and 10 minutes of sample preparation. So that got us pretty excited. We then tested this method out on a bunch of different patient samples, which I'm sort of showing you here, using both the colorimetric readout as well as in-tube fluorescence. And overall, the results were pretty good. We saw 94% concordance overall with RT-PCR, which is sort of the gold standard approach here. The method was very specific. There were no false positives at all. There were still a few samples that we didn't pick up, and this is definitely something that we are continuing to optimize as we work on the method. The overall assay time was really quite fast from sample to answer, a total of 50 minutes. And there's no nucleic acid extraction or anything required for this method. So we're quite excited about SHINE as we think it really fills a gap in the spectrum of tests that are available as far as being fast and relatively easy to perform. And in the future, we're really excited for its potential going forward to really allow testing to be performed in a wide variety of places that it can't currently be performed. But that's really only one part of the testing problem, which is being able to perform tests anywhere. there's a separate issue, which is that in general, we are not performing nearly enough tests for nearly enough things. And what do I mean by that? Well, really, another way of thinking about this detection problem is to plot the problem on two axes. One is multiplexing the number of different targets we might want to detect at a time, and the other is throughput, the number of samples that we might want to be able to test at a time. And for different applications, there are different sort of needs in terms of throughput and multiplexing. And in particular, in an outbreak situation, you might not need to test for as many viruses, but you're going to want to test loads and loads of samples. Whereas if you've got, say, an infected patient, you'll want to test for lots and lots of things if you don't know what is making them sick, but there might not be as many patients to test. And we think disease surveillance is really something that falls in between and is an important application here. Most existing technologies, though, unfortunately, are really built around this paradigm of testing a small number of samples for a small number of things. And while that is useful and does work, I think we really want to move up and to the right on this graph. And so that's really what we set out to do. Building very much off of SHERLOCK, which I'm sort of showing you an overview here. SHERLOCK is really about taking a single sample, amplifying it, and then detecting a specific nucleic acid sequence present in the sample using Cas13. And our colleagues in Feng Zhang's lab had shown that you can do 4-plex multiplexing by using different Cas12, Cas13 orthologs, but we got a little more ambitious and said, well, what if we could really multiplex out to the hundreds and do this sort of testing on lots of samples at once? We really wanted to scale up our ability to perform nucleic acid testing. And I think now more than ever, that need is quite evident, as it takes a long time to get test results back at the moment. And we're typically only ever testing any one person for one virus at a time. And that's really, really not ideal if you don't know what somebody has. So we developed a technology called CARMEN to try and address this problem in close collaboration with Paul Blaney's group here at Broad. And CARMEN stands for Combinatorial Arrayed Reactions for Multiplexed Evaluation of Nucleic Acids. And what it really lets us do is miniaturize these Cas13 detection reactions, so we can perform lots of tests in parallel, thousands at a time, and it simultaneously reduces the reagent consumption. And what's particularly neat about the technology is that it really lets us test lots of different samples against lots of different CRISPR RNAs at a time and really thoroughly check to see what viruses may or may not be present in a sample. And so how does it work? The first step is miniaturization. So we take amplified samples or detection mixes and we color code them by mixing different ratios of fluorophores in them. And then they get emulsified to form individual sets of droplets that are labeled, so we can tell sample 1 apart from sample 2 and detection mix 1 apart from detection mix 2, which will have different CRISPR RNA sequences in them. And we can then pool all of those droplets together and load them onto a microwell array. And that array will organize the reactions into pairs. And in particular, each pair can contain, for example, 1 sample and 1 detection mix. And so it basically forms all of the different pairwise combinations of samples and CRISPR RNAs. We can then image that microwell array to tell what is where. And so that tells us which sample and which CRISPR RNAs we are pairing up. We can then use an electrical pulse to merge the droplets and initiate the reaction and read out the fluorescence over time, again, using a microscope for readout. And once we analyze the data, that really gives us this picture of what particular viruses, say, may or may not be present in a large set of samples. And this method works quite well. Actually, when we originally developed it, Zika was the virus that everyone cared about. So we initially demonstrated that we could detect Zika using CARMEN. And what you can see here is sort of an example of what our data looks like. We get a distribution of fluorescence values. Here, I'm just doing a dilution series of Zika cDNA, and you can see that the method is quite sensitive. And you can see that, for example, at any given concentration, there is a distribution of fluorescence values that we get. What we typically do here is we take the median of that distribution and we show it on a heat map. So you're going to see a lot of heat maps going forward, and that's what I'm showing you is the median of that distribution. Here it's just a 1 row heat map, but don't worry, they're going to get a lot bigger soon. We also did a comparison side by side with SHERLOCK on a plate reader and showed that the performance was similar between the two methods, which was really a nice reality check that things were working well. So now I want to talk about a couple of different applications of CARMEN. We can use it to detect different species, subspecies, and SNPs. So I'll start with species, and here we used a design software developed by a graduate student, Hayden, in the lab called ADAPT to design and test a set of CRISPR RNAs against all human-associated viruses with at least 10 available genome sequences. So we set ourselves out to try and see if we could do that and be successful. And at the time that we did this, there were 169. I can tell you right now that there's at least 170 because one of them is SARS-CoV-2, which was not on the list at the time. This was back in 2018. And so then we comprehensively tested the cross-reactivity of all of these different designs in a giant 169 by 169 matrix using synthetic targets at a high concentration to really ask the question, is this technology going to be specific enough to allow us to do multiplex testing at this scale, which is a real challenge because many methods have cross-reactivity between different sequences, and that would really pose a challenge and make it difficult to interpret any results. Unfortunately, as you can see here, it worked quite well. What I'm showing you is again a heat map. On the x-axis, I'm showing you all of the different targets we tested. The y-axis is all of the CRISPR RNAs arranged by family. This includes every virus that you've heard of and almost certainly a number that you haven't. And of those CRISPR RNAs, 94% were specific and had an on-target signal greater than our threshold. This was after one round of optimization, so the technology doesn't work perfectly, but in general, we can now make designs that work quite well. On average, their performance is very high with a median area under the curve of 0.997. So we took this panel and then we tested it on patient samples. And what I'm showing you here are just some results of that testing, where we compared the results of CARMEN to the results of next generation sequencing. And what we saw was really high concordance overall. For any given amplicon, we found that CARMEN was more sensitive. So if we asked the question, does the CARMEN amplicon exist in the sequencing data? Then we found a higher sensitivity for CARMEN, but the advantage of NGS is that it can look for the entire genome. And so there were some cases where what we saw was that some other part of the genome was present, but not the part of the genome we were trying to detect. And so overall, the sensitivity was therefore similar. There were about an equal number of cases where CARMEN detected it, but sequencing didn't, or vice versa. But I think the big advantages here are that the turnaround time of CARMEN is much faster, so we can run a CARMEN experiment within a single day, and the cost is a lot lower. We can also do subspecies identification, and here we chose the subtypes of influenza, H1 through 16 and N1 through 9, and tried to design panels that could differentiate H1 through 16 and N1 through 9. And I think here, the data really speak for themselves. We can basically do that successfully, again, using synthetic targets. On the left of the slide, you can see H1 through H16, sorry, and N1 through N9. And on the right, a set of patient samples that were either H1N1 or H3N2, graciously provided to us by the CDC, which showed perfect concordance with both RT-PCR and sequencing. I unfortunately don't have time to show you the results of the SNP discrimination, but suffice to say that we can use CARMEN to discriminate SNPs. And in particular, we've designed assays against 27 different HIV drug resistance mutations and tested that out on clinical samples. And if you want to read more about that, you can definitely look at the paper where we go into lots of detail about that testing. So now I just want to summarize and remind everyone that CARMEN works quite well for detecting species, subspecies, and SNPs. And this was really exciting, and it was really great to see our paper finally come out earlier this year. But we have been hard at work to take CARMEN and apply it to COVID-19, and I'll just spend the next couple of minutes going over that in a little bit of detail. And this was actually quite fortunate because last year we had decided to see if we could design a CARMEN panel against a set of respiratory viruses, because we've had a long-standing interest in those viruses, and in particular, started a collaboration with the CDC last fall to focus on influenza. And so what I'm showing you here are the results of testing that panel. And you can see we've added SARS-CoV-2 as well as a number of other human coronaviruses to the panel, along with a set of other respiratory viruses. And what you can see here is the performance is quite good. Many of the viruses are detected quite sensitively with CARMEN. And we've also tested this out on patient samples and see really good performance there as well. On this particular set of 10 patient samples, we're seeing 100% concordance. When we test a larger set, it's not quite at 100%, but the concordance is really very, very high. And I think that leverages a lot of the sensitivity of Cas13, and it's been really exciting to see that assay get developed and really start to get used more widely. So now I just want to take a moment and bring this back to where I started, which is really this challenge of developing COVID-19 testing technologies that scale. And so on these same axes that I showed you before, our goal is really to develop technologies that can test hundreds to thousands of samples with a turnaround time of less than an hour, and really ideally 30 minutes or even 15 minutes. And I would say we're still a ways from getting there. In red, I'm showing you a bunch of existing technologies that are attempting to get at this problem, but in general, they fall short either because it takes a very long time for turnaround, and that's especially true for methods like sequencing and RT-PCR. There are other methods that are much faster, like the Abbott ID Now or Cepheid Gene Expert. Unfortunately, those methods are really designed to only test a single sample at a time, and so it's difficult and indeed often cost prohibitive to scale them up to test lots of samples at a time. And so we've been working on this problem sort of from two angles. The first is really taking technologies like CARMEN, which I just described, and trying to move them to the right on this graph. So CARMEN is something that can test lots of samples at a time, but at least in its initial implementations required about 6 to 8 hours of turnaround time. And our goal is definitely to move that to the right. And sort of similarly, we think a lot about these more point of care technologies, things like SHERLOCK and RPA, and really trying to take those technologies and make sure that those can be scaled up to test lots of samples at a time, because many of them have really been optimized to test small numbers of samples with a fast turnaround, and that is helpful, but the size of this problem is ginormous. And ultimately what we're really going to need is all of these technologies working together in concert if we want to be successful. So it's really important, I think, that people are taking different approaches because as we've seen, the demand for testing is really high, and I think is only going to increase in the fall for a variety of reasons. And with that, I just want to thank a really wonderful and large team of people who have helped make this work possible, including a number of wonderful graduate students and postdocs in Pardis Sabeti's lab, as well as RAs, our collaborators in Paul Blaney's lab who really helped develop the CARMEN technology, as well as some folks in Deb Hung's lab, collaborators at CDC who provided us some samples and funding. Thank you.
Great, thank you, Cameron and Jacob, again, for those talks. It's a great representation of the work, the tremendous work, really, that's been going on in the Sabeti lab and with all of our partners over the course of the pandemic. And I think we're all, Jacob, Cameron, and I are all just really conscious that while we were mostly working from home, there was a small army of folks who were going into the lab every day to make this work happen and putting themselves at risk to do this work. And so big shout to the lab teams who have been underpinning the stories that Jacob and Cameron have told you today. And so now we have time for a few questions. I want to welcome Pardis to the room here to take any questions that you may have. We've been trying to keep up with a few questions in the chat function there, but keep them coming, and we'll get through as many as we can now. So let's start with a question to you, Cameron, just picking up on kind of where we are in the real world for getting SHERLOCK and CARMEN out. Can you tell us anything about the path to EUA approval and real kind of implementation of these technologies?
Yeah, it's definitely something we're really excited about. I think it's something that, you know, previously I would have told you, oh, this is something that's a really long ways off, but I don't think that's the case. There's a lot of excitement around this, and it's something we're actively, you know, working to pursue. And my hope is that in particular, something like a CARMEN respiratory panel could become available for people to use. We see a great need for that emerging in the fall and winter months as other respiratory viruses start to circulate again. And I think there's going to be a lot of lingering questions around, if someone gets sick, do they have the flu? Do they have SARS-CoV-2? Do they have something else? So we're really definitely moving full force towards that.
Great, thanks. And there's a question, there's been a few questions probably to Jacob about the mutation rate of SARS-CoV-2 and how the implication of that for what we can say about the data in terms of the genomic epidemiology, and maybe also, Cameron, you can speak to it in terms of diagnostic performance.
So the mutation rate is consistent with other coronaviruses and other positive sense RNA viruses. Substitution rate of the virus is approximately 1 × 10⁻³ per site per year. So it is a pathogen that is involving roughly on the same order of magnitude in which it's spreading, and we can use that information to draw inference about the timing of spread. It's a relatively noisy molecular clock, so there is some uncertainty. That uncertainty is reduced by considering larger sample sizes. There are definitely concerns about substitutions in the primer sites, primer binding regions of diagnostic assays. There's at least one that's been reported in the WHO assay. Fortunately, the mutation is quite rare, but it definitely does decrease the sensitivity of one of the targets. That's actually the assay that does the bulk of the testing at MGH, so we've been monitoring closely for that variant, haven't seen anything. We've done an extraordinary amount of sequencing, but I actually think we're under sequencing. So we're trying to do more sequencing because we just aren't nearly anywhere capturing the diversity of the tens of millions of human infections that are out there.
Yeah, Cameron, did you want to elaborate on that with respect to diagnostics?
Yeah, sure. I mean, it's definitely something we think about a lot from the diagnostic perspective. We want to make sure we're trying to design to regions of the genome that are not evolving as rapidly, but sometimes that does happen. And I think one of the nice things about our technology in particular is that you can relatively easily redesign a CRISPR RNA or a primer if needed. And we've demonstrated that in the past. If a new mutation emerges, for example, we can even make an assay to detect that mutation specifically within a week.
Awesome. There have been a few questions, Jacob, about the sampling framework in general for the genomic epi work, and in particular with respect to the D614G populations. Do you want to speak to that, just what we can say about that in our data?
Yeah, so let me just, so there are a couple of things. One about the sampling proportion and about the sampling type. So I want to clarify that we do not have a random sample. So we have to be careful about how we generalize from the sample of genomes that we do have. But I think it is representative, particularly of the early known cases in the state. Overall, we've sequenced about 1% of the reported infections. The true number of infections, I think, is unknown. This is within Massachusetts. So I think we need to do more, and we need to ensure that we have representative samples that will allow for more generalizable inference. There's been some questions about D614G, and I would just emphasize that this is an incredibly exciting but also controversial area. The Korber paper generated a lot of controversy when it was shared as a preprint. It's now through peer review and available in Cell. The effect seen there does suggest increased viral loads with the derived variant, and there's some infectivity data using pseudotype virus, both in the Korber paper and in the Yurkovetsky et al. Manuscript on bioRxiv from Jeremy Luban's group. But this remains a preliminary, intriguing result. And there have been comments and concerns about potential confounders, including secular trends of changes in virus load over time during the course of the epidemic. I would say we share those wholeheartedly. We're trying to strike a balance between trying to investigate this area, but to do so in a way where we're really confident in the findings. And I would say that I put it in there because this is a internal meeting. I think it's not ready. We don't feel it's ready to put up on the website. But I think this is an area of ongoing interest and potential importance that requires further investigation.
Right, perfect. So there are a lot of great questions in the chat here. But before we wrap up, I just want to give a chance to hear from Pardis, since, as Jacob mentioned, the fact that we were able to respond to the COVID-19 crisis as we have as a group is really kind of built on the foundation of the work that Pardis has been leading for a decade, both globally and at home. And I think these are two pillars of the type of work we do in terms of the epi and the diagnostics using genomic data, and just wanted to hear Pardis's thoughts on where we're headed with this in the future, what we've learned, where we can take it, and how it all kind of comes together.
Oh, wow. All right.
No pressure.
Yeah. Well, I mean, yeah. So I should say this is, as a faculty at this point, your main job is just being a talent manager, and this is all possible because of the talent here on this panel and in the deep set of individuals that Bronwyn mentioned, grad students, techs, staff scientists, and beyond that make this work possible. And grateful to all of you. I just want to make a couple comments before I'm giving myself time to answer that question. To also say thank you to the so many people that are here in late July listening to this, and already have been hearing from folks to collaborate. Excited about that too. As far as where we're going, I mean, we're just head down trying to stop COVID in every way that all of us can, but it has accelerated so many of our activities. We had these sort of long plans of getting these diagnostics out sometime in the future, and now everything's trying to go to EUA as fast as possible. So those moves and trying to really ramp up our sequencing capacity as we can to be able to continue to track this. And a lot, this work has really been driven by the people, by Bronwyn, Jacob, and Cameron, and I've been doing some side gigs, working with some really trying to build up our ability to track these viruses, even faster, as well as educate the public. And so I think for like a larger set of things that we're really interested in, we just kind of came together. We won a prize called the Audacious Prize to build something called Sentinel. But it's really bringing together all these different pieces of detecting viruses, tracking them, and building ways to find them wherever they occur, and to use a lot of different information tools like Viral NGS and the Terra platform that Jacob talked about, but also we've got other dashboard visualizations and mobile applications that we've been developing over the years to be able to much more quickly identify viral threats. And then lastly, from that education standpoint, we really want to empower every actor in the system. And so that's why we've invested in education. We've trained about 900 individuals from around the world, like deep courses, month-long courses, multiple month-long courses in diagnostics, genomics, bioinformatics. And probably one of the projects most near and dear to my heart right now is something called Operation Outbreak, run by Andres Klubri, which is a deep curriculum for students of all ages, but we've done a lot of 7th and 8th graders, K through 12, high schoolers and college students in really understanding epidemiology, statistics, bioinformatics, all of the pieces that you have, history, government. I mean, there is no education, there's no existential threat to society that kind of touches on so many different aspects of biology, stats, and also affects our daily lives. We have flu outbreaks every year. And so we have this education program we're rolling out that's paired with a mobile application that spreads a virtual virus via Bluetooth. Right now, actually starting tomorrow, about 2,000 students in the Chicago public school system are going to be doing a virtual outbreak using this Bluetooth that actually spreads a virus at 10 feet apart. So they can, even if they come close contact, they can see what would have happened if they were closer and get to see how this all could unfold and think about how to stop it better. So, you know, all the different people, we have lots of different things cooking
going on, right?
And collaborating with lots of different folks on it. But sorry, that was a little bit long.
No, that's perfect. Thank you. So this is ongoing work. We are still working hard on all of this and look forward to updating you in the fall after a well-deserved summer break for the organizers of the IDMP seminar series. I just, on behalf of our whole community, want to take the opportunity to thank the organizers and just the extraordinary vision and leadership of Deb to put this whole series together. I think since it launched, just, you know, as COVID hit Massachusetts, this has been such a highlight in my calendar, and I'm sure for many of you as well, and just a great resource to the community to learn together in real time on key issues. And we're super proud to have been part of it and to close it out for the summer today. So thank you, Deb and Romnick. Thank you to all the presenters in this series, and thanks everyone who tuned in this morning, and we'll look forward to seeing you all in September. Thanks, bye for now.